My name is Aurora, and I'm a PhD candidate in the Salinas lab in the biology department here at UNM. So when the COVID pandemic hit, I felt like I had already begun this really amazing model called the zebrafish, and so we decided to jump in and try to use the zebrafish as a model for SARS-CoV-2 in the nose. There was this interesting thing that happened to a lot of people who became sick with the virus, is that they lost their sense of smell.
In my lab, we are really interested in the interactions between viruses and the olfactory system. So why do we lose smell when we get infected with a virus? Zebrafish are really beautiful small fish that are widely used in biomedical research, and also they are transparent, so we can do a lot of things in terms of imaging the whole animal. And they're also very good to manipulate genetically, so we can tweak things in the zebrafish to change the genetic composition. So let's say our collaborators in France are making a humanized zebrafish that expresses the receptors for COVID-19 virus like human receptors, so that the animal will get infected similarly to how it gets infected in us.
Something that is becoming better understood in the last 10 years is that neurons, which are really fast communicators all the way across your body, are actually interacting with your immune cells, which are thought to be a slightly slower response, and that these immune cells in turn are giving cues to your neurons. And no one so far has looked at how these neuronal and immune signals are communicating in your nose. So with the pandemic, we thought with so many people getting infected, this was a really important thing to study, and how the virus is driving your neurons to detect the virus as it's there and replicating in your epithelial tissue in your nose, and how your immune cells are protecting you. So we thought we had a really cool model, not just to science, but also the interesting part in how neurons and immune cells could be working together to protect our brain, which is one of our most vulnerable and most important organs as humans. We, in the adult model and here in the biology building, use the spike protein from the SARS-CoV-2 virus. I can put it directly into the little fish noses and see how the nose and the neurons that sense smell responded to the virus. And then I did some histological stains and saw that the neuroepithelium, or the area where the sensory neurons that detect smell live, was really extremely destroyed. This is translatable to humans because all of our SARS-CoV-2 or COVID-19 vaccines that we're all taking are based around the spike protein. If you've seen in the news those pictures of the virus in the ball, they always have these little knobs around it, and it kind of looks like a crown, which is why it's called a coronavirus. And this protein that creates the knobs is called the spike. If a cell were to bump into this virus, the first thing it would come into contact with would be this spike protein, because it's the thing protruding up. But we don't know what this protein by itself can do. It interacts with a receptor, and that's how it gets into the cell. So understanding how it could affect the immune state of your brain, how it will change your sense of smell by itself, is really important and translatable. Right now, we're the only ones asking that question on a model that is so genetically tactable, pliable. Hopefully it can help with perhaps long COVID symptoms and understanding the effect of vaccines on the population.
We are very excited because we think that there's a lot of potential, for instance, to use this model to see how smell recovers. Perhaps this will be a great model to see, if you do A, B, C, then your smell comes back faster because the neurons regenerate quicker. So that's some of the applications that we are thinking of, and we can do it without using the infection, with a very cheap animal model, and very quickly to generate data.